All right, today we're diving deep, like microscopic deep, into something absolutely fundamental to life itself, the plasma membrane. You can think of it as this tiny, super intelligent border that literally decides what's life and what's, well, not. It's the gatekeeper that makes everything else possible, and we're going to see how.
Okay, here's a weird one for you. What do you, a plant, and a pickle all have in common? I know, it sounds like the setup to a really bad joke, but the answer is actually pretty amazing. Believe it or not, they all depend on this tiny microscopic border guard to survive. We're talking about the plasma membrane. Every single living cell, yours, a plant's, even the little microbes that turned a cucumber into that pickle, they all rely on this little gatekeeper to control what gets in and what gets kicked out. It's the key to the whole operation of life. So let's start with the big picture. What is this border's #1 job? Well, think about it. A cell has to eat, right? It needs to bring in nutrients, get oxygen, and at the same time, it's got to take out the trash. So how does it manage all that traffic without turning into total chaos? The secret sauce is a property called selectively permeable. It's a fancy term, but the idea is simple. It's like a really, really good bouncer at a club. You know, some things, the good stuff, get waved right on through. But other stuff? Nope, you're not on the list. And it's this choosiness that lets the cell keep its inside world perfectly stable and totally different from the craziness outside. Okay, so how do you even build something like that? A barrier that's tough enough to hold a cell together, but also flexible and smart enough to run that bouncer operation. Let's take a look under the hood and meet the parts. The model scientists use to describe this is called the fluid mosaic model, and honestly, the name is perfect. Fluid because nothing is stuck in one spot. All the little pieces are constantly shifting and flowing around, kind of like buoys in the ocean. And mosaic because it's made up of all these different bits and pieces, lipids, proteins, cholesterol, all working together to create this amazing functional pattern. So the main foundation, the wall itself, is something called a phospholipid bilayer. It's genius, really. Each molecule has a head that loves water and a tail that hates it. So what do? They automatically line up in 2 rows, with all the water-hating tails tucked away on the inside, protected from the water on both sides of the membrane. This creates this perfect self-building fence that separates the watery inside of the cell from the watery outside. But just having a fence isn't enough, right? You need gates, and that's where the membrane proteins come into play. Some of them, the channel proteins, are basically just tiny tunnels. Little ions and things can just zip right through. But others, the carrier proteins, are more like a personal ferry service. They literally grab a specific molecule, change their shape, and carry it across to the other side. Pretty cool. And to finish off the structure, you got a couple more key players. First up is cholesterol. It kind of wedges itself in between all those phospholipids and acts like a stabilizer. It makes the membrane stronger, but also keeps it flexible. Then you have these things called glycolipids and glycolipids. The easiest way to think of them is as the cell's ID tags. They stick out and tell the immune system, hey, I'm a friend, and they also act as docking stations for important messages, like hormones. All right, so we've got our border built, we know what it's made of. Now, what are the rules of the road for getting across? It actually boils down to 2 main ways. You can either go with the flow, or you can fight against it. And this is the core difference right here. Passive transport, that's the easy way. It's like floating downstream on a lazy river. It takes 0 energy, because you're just naturally moving from where there's a lot of something to where there's less of it. Active transport, though, that's the total opposite. That's trying to swim upstream, against the current. And to do that, you're going to have to burn some serious energy, which for a cell means using ATP. Now, even that go with the flow, passive transport, has a few different styles. First, you've got simple diffusion. This is for tiny molecules like oxygen and CO2 that can just sneak right through the lipid fence, no problem. Then there's osmosis, which is just the special name we use when we're talking specifically about water moving across. And finally, you have facilitated diffusion. This is for stuff like glucose that's a bit too big or clunky to get through on its own. It needs a little help, so it uses one of those protein gates we mentioned. So let's just break it down one more time. Passive, no energy needed, just follows the natural gradient until everything's balanced. Active requires energy, goes against the gradient using special protein pumps, and it's what lets a cell hoard the good stuff or actively kick out the bad stuff. It's a game changer. Okay, now for the fun part, the drama. What happens when these rules, especially osmosis, get pushed to the limit in the real world? Well, this is where things can get, let's just say, a little explosive.
So, picture this. We take a single red blood cell and drop it into a beaker of pure water. Now, this is what we call a hypotonic solution. Basically, there's a lot more pure water outside the cell than inside, and the laws of osmosis are clear. Water always moves from where it's crowded to where it's not. So, water's going to flood into that cell, trying to even things out.
And what happens? The cell starts to swell up like a balloon. It gets bigger and bigger until, pop, it bursts. The plasma membrane just can't take that much pressure on its own. It's a catastrophic failure, and it has a name, hemolysis. Okay, but what if we do the exact same experiment with a plant cell? Same pure water, does it meet the same explosive end? Nope, it swells up, for sure, but it doesn't burst. And why not? Because plants have a secret weapon, a super tough, rigid cell wall right outside their membrane. So as the membrane swells and pushes out, the wall pushes right back. That internal pressure is what we call turgor pressure, and it's literally what keeps plants standing up nice and tall.
All right, let's flip it. What if we put both cells in a super salty solution, a hypertonic one? Now the situation is reversed. There's more water inside the cells than outside, so water rushes out. The poor animal cell just shrivels up like a raisin. We call that crenation. The plant cell also loses water, but its strong wall stays put. Instead, the membrane inside just pulls away from the wall, which looks pretty wild. That's called plasmolysis.
Okay, so why should you care about shriveling cells and turgor pressure? Because this microscopic drama is happening all around us, all the time. In your own body, on your dinner plate, everywhere. Think about it. The very oxygen you're breathing right now, it gets into your blood through simple diffusion. That lunch you ate, your gut is using active transport to pump all those valuable nutrients into your bloodstream. And your kidneys are constantly using osmosis to pull water back into your body to keep you from dehydrating. This isn't just textbook stuff. It's happening billions of times a second just to keep you alive.
And hey, we finally get back to the pickle. The whole reason pickling works, or salting fish, or making jam, it's all osmosis. You create this super salty or sugary environment, and any poor little bacterium that lands on it gets all the water sucked right out of it. Spoilage stopped, just like that. And it's the same reason why if you dump way too much fertilizer on a plant, it wilts. You've made the soil hypertonic, and it starts pulling water out of the roots instead of letting it flow in. And this stuff is huge in medicine and technology too. You know when someone gets an IV drip in the hospital? That saline solution is isotonic. It's perfectly matched to the salt concentration of your blood so it doesn't mess with your cells. And you've probably heard of reverse osmosis for water filters, right? That's just us copying the cell membrane, using pressure to force pure water through a filter and leave all the gunk behind. So you see, from a single cell all the way up to global technology, this tiny, smart border is the unsung hero, the quiet gatekeeper of life. It shows us that at every single level, survival depends on having a healthy, functioning boundary, which kind of makes you think, right? If one little cell falls apart when its border fails, what happens when the bigger borders, the ones protecting our planet's resources, start to break down?